The overall goal of this procedure is to promote remyelination and clinical improvement in animals with established demyelination via transplantation of neural stem cells into the spinal cord of JHMV-infected mice. This is accomplished by first preparing the animal. The second step of the procedure is to perform a laminectomy at thoracic vertebrae 10. Cells are then injected into the exposed spinal cord. After a successful injection, the wound is closed. Ultimately, presence of mouse neural stem cells in the spinal cord parenchyma can be visualized by methods such as fluorescent microscopy.
Neural stem cell transplantation into the spinal cord of JHMV-infected mice can help answer key questions into the field of cell replacement therapies for the treatment of demyelinating diseases, such as how the cells migrate, proliferate, and differentiate. Also, how the cells affect the central nervous system, and if we can modify the cells or the spinal cord environment they're transplanted into to enhance their therapeutic benefits.
Although this method can provide insight into cell replacement therapy in a viral model of demyelination, it can also be applied to other models of demyelination and injury, such as experimental autoimmune encephalomyelitis, EAE, and spinal cord injury.
Before preparing cells for injection, prepare all necessary solutions. Next, clean and sterilize all equipment. Prepare the surgical area and set up the micromanipulator. To prepare the cells for transplantation, trypsinize and resuspend them in a 50 ml conical vial. Wash the cells to be transplanted 3 times in HBSS. Count the cells before the final spin. After the final spin down, decant HBSS and leave